Hi everyone, my name is Sophie Farrow and I'm based at the Liggins Institute in Auckland, and today I'm going to be talking about our recent paper published in Brain titled Establishing Gene Regulatory Networks from Parkinson's Disease Risk Loci. So in 2019, the largest genome-wide association study for Parkinson's disease was conducted by Mike Nels and colleagues, and together they identified 90 independent variants across 78 genomic regions that are associated with the risk of Parkinson's disease. And these variants identified in the study are single nucleotide polymorphisms, or SNPs, and they essentially are just one base in the DNA changing at any time. The identification of these 90 variants of course sparked the question as to how these risk variants are actually contributing to the development of Parkinson's disease. And in theory this question sounds relatively simple to answer. Yet the vast majority of these 90 risk variants are actually located in non-coding regions of the genome, which makes assigning function to them extremely hard. Here within our lab we developed a pipeline to begin to answer this question. So the pipeline integrates different datasets, including population-level genotype data, and also data on the spatial organization of genome. Now integrating the spatial data has enabled us to identify 3 different types of SNP gene interactions, or EQTLs. So we have cis, where the SNP and gene are within 1 megabase of each other on the same chromosome, and this is fairly similar to the nearest gene approach, which is often taken. And then we have trans-intrachromosomal, which is when the SNP and gene are on the same chromosome, but much further away, likely more than 1 megabase from each other. And finally, trans-interchromosomal, when the SNP and gene are on completely different chromosomes, but a regulatory interaction occurs due to the 3-dimensional organization of the genome. So using this approach, we found that 76 of the 90 SNPs were associated with the expression of 518 different genes across 49 tissues. And intriguingly, we found that a third of these regulated genes were actually identified as a result of long-distance regulatory interactions. And one particular connection which is quite interesting is between the SNP RS108-47864, which is located in the HIP1R gene on chromosome 12. And this SNP is regulating the expression of SYNJ1 located on chromosome 21 in the brain cortex. Now SYNJ1 has already been implicated in familial Parkinson's disease through ancestral studies, but this is the first time that SYNJ1 has been associated with the sporadic form of the disease. So we then asked as to whether these SNPs are likely acting as part of a network, and to answer this we used the Leuvian clustering algorithm to investigate the 518 genes regulated by the 90 SNPs. Using this approach, we identified 9 different clusters which were enriched for different biological pathways, such as cluster 5, which is synaptic vesicle recycling, and cluster 8, which is autophagosome assembly. And we argue that these different clusters may be a potential method for stratifying patients, with different pathways contributing disease development, depending on which SNPs are present within the individual. And it's also important to note that the little orange circles represent genes that are regulated through long-distance mechanisms. And as you can see, in many of the clusters, without these genes, the clusters would fall apart. And so it's important to consider both near and far gene connections. We then considered whether the SNPs have a regulatory effect in fetal cortical tissue to see if there could be a potential neurodevelopmental effect of these SNPs. So as you can see here on the left, these set of genes are regulated by some of the 90 SNPs, but only in the fetal cortex and not in any adult brain tissues at all. We also found a subset of genes that are regulated in both adult and fetal cortex, and then another subset of genes which are regulated in the fetal cortex and non-cortical brain regions in adults. So this suggests that there is a likely neurodevelopmental effect of some of the SNPs for Parkinson's disease. So I guess the final question to ask is, what is the real-life impact of these findings for the future of Parkinson's disease? The results presented within this paper provide new potential avenues to consider for the stratification of idiopathic or sporadic Parkinson's patients. And we also identified different pathways which may be considered for future therapeutic targets. We went on to explore which genes and pathways are affected across a number of different tissues, and we argue that this differential involvement of different tissues in different individuals may explain some of the phenotypic variance and heterogeneity that we see in Parkinson's individuals. We also highlight the importance of considering both cis and trans dysregulation of gene expression, which is consistent with predictions from the omnigenic model of complex disease, which is likely applicable to Parkinson's disease. Of course, the research conducted in this paper is only a small part of a big jigsaw to achieve precision medicine in Parkinson's disease. Our ultimate goal is to identify the causative pathway or mechanism for each individual and be able to treat them accordingly. And we hope that this work moves us a tiny step closer to the stratification approach. Thank you for listening, and I hope you enjoy reading the paper.